Okay, let's unpack this. It's funny how certain words just become like permanent fixtures in our vocabulary.
Right, to the point where we just assume we completely understand them.
Exactly.
Yeah.
You hear HIV or AIDS, and your brain instantly fills in this whole narrative. But when you actually dive into the comprehensive biological and medical notes we're exploring today, you realize most of what we think we know is really just scratching the surface.
It really is. It requires a pretty profound shift in perspective, moving from that standard public health narrative we all know to the actual, cellular reality of it.
Yeah, because today's deep dive is all about decoding this master of biological disguise. We're looking at exactly how this virus outsmarts the human body on a microscopic level and how modern science is finally fighting back.
Because it stops being just a medical condition, right? It starts looking like this highly coordinated, almost impossibly sophisticated heist happening right inside the bloodstream.
A heist. That is the perfect way to look at it. Because, well, the very first thing we have to clear up is a massive misconception. You might think HIV and AIDS are basically synonymous, like just two interchangeable words for the exact same thing.
Which they absolutely aren't.
Right. They aren't. HIV is the virus itself, the physical microscopic invader. AIDS is actually just the final severe crisis stage of this long, drawn-out cellular war that can take, what, a decade to unfold?
Yeah, up to a decade. Confusing the two is kind of like confusing a termite with a collapsed house.
Oh, that's a great analogy.
Right. The Human Immunodeficiency Virus, HIV, is the termite. The Acquired Immunodeficiency Syndrome, AIDS, is the structural collapse that happens years later when the hidden damage is just too much for the framework to bear.
So before we can even talk about a house collapsing, we have to look really closely at the termite. What does this invader actually look like, and where did it come from? Because here's a detail a lot of people miss. There isn't just one type of HIV.
No, there are two main strains fighting this war, and their origins and behaviors are completely different.
Which is wild to think about.
It is. So HIV-1 is the strain most people are familiar with. It's responsible for about 95% of cases globally. It's highly infectious, progresses very quickly, and originally crossed over into humans from chimpanzees in Central and West Africa.
Okay, so that's the aggressive one.
Right. Then you have HIV-2, which is largely confined to West Africa. It progresses much slower, is far less infectious, and actually originated from a completely different primate, the sooty mangabe.
Regardless of the strain, though, the physical structure of this thing is insane. We're talking incredibly tiny. It's an icosahedral structure, so basically shaped like a microscopic 20-sided die from Dungeons & Dragons. And it's only 100 nanometers across.
Yeah, to put that scale into perspective, you could line up about 10,000 of these viral particles across a single millimeter.
10,000.
Yeah, it is practically a ghost.
And the way it's built, I kept picturing like a stealth submarine. Because it doesn't just have its own outer shell, it literally steals its camouflage from the human body.
It does. When a newly formed HIV particle exits a human cell, it rips off a piece of the cell's own membrane and just takes it with it.
Steals its jacket on the way out.
Exactly. Specifically from areas called lipid rafts, which are thick with cholesterol, so it's pulling this biological catch me if you can while wearing our own uniform.
That's...
Swimming around the bloodstream draped in human cellular material is just a remarkable evolutionary adaptation. And sticking out of that stolen lipid envelope are these external spikes, 72 of them to be exact.
And those are the grappling hooks, right?
Yes, they're specialized glycoproteins, the most important one being GP120.
Okay, so that GP120 protein sits on the outside of our stealth submarine. And its entire job is to seek out and dock onto very specific ports on our white blood cells, the CD4 receptors.
Right, those are the targets.
Once it locks on, it breaches the hull. And inside that stolen envelope is a bullet-shaped core, the capsid, made of P24 protein, I think.
You got it. And inside that core is where the actual payload sits, which is the virus's RNA and some extremely specialized enzymes.
Okay, and the enzymes are where the real trick happens.
Yeah, what's fascinating here is the nature of that genetic payload. HIV is a retrovirus, which means it completely defies the normal rules of biology.
Because normally, in every human cell, the flow of genetic information goes one way, right?
Yeah.
DNA makes RNA, which makes a protein.
Exactly. That's the fundamental dogma of biology. A blueprint makes a copy, the copy builds the house, but the virus carries an enzyme coded by the pull gene called reverse transcriptase.
Oops.
Just like the name implies, it completely reverses that normal flow. It reads the virus's RNA and translates it backward into DNA.
That's just completely backwards. It's like the blueprint is rewriting the architect.
That's a perfect way to put
It
it.
translates its own alien blueprint into human biological code.
Right.
So the submarine docks, drops this payload, and immediately starts reverse engineering itself. Which brings us to the actual heist. Once it's inside, how does it pull off the takeover?
Well, it executes a sequence of incredibly precise steps. First is attachment, where that GP120 grappling hook binds to the CD4 receptor on a human immune cell.
Right.
Next comes fusion. The virus's stolen envelope merges seamlessly with the human cell's membrane, dumping that bullet-shaped core inside. Then the core uncoats, releasing the RNA and that reverse transcriptase enzyme.
So RNA becomes viral DNA. But having a loose strand of viral DNA just floating around the cell isn't enough, is it?
No, it has to get into the command center. So another viral enzyme called integrase grabs that newly minted viral DNA, carries it straight into the nucleus of the human cell, and literally splices it into our own chromosomal DNA.
It just cuts our DNA open, glues itself in, and stitches it back up. That is terrifying.
It is. And from there, it seems like the virus has two completely different strategies. It can go loud or it can go dark.
Right. Active versus latent. So in an active infection going loud, it essentially hijacks the cell's machinery. It forces the cell to mass produce thousands of new viral copies, which then bud out, steal more of the membrane, and go off to infect others.
Exactly. But the alternative strategy is latency, and that is what makes HIV so uniquely devastating.
Why is that worse?
Because in a latent infection, the viral DNA integrates into the human chromosome and just stops. It becomes what virologists call a provirus. It doesn't produce new viruses, it doesn't destroy the cell, it just sits there, completely dormant.
A hidden piece of alien code within the host's own DNA. And this is where it targets macrophages, right? Because macrophages are basically the garbage trucks and transport vehicles of the immune system.
Right, the cleanup crew.
So HIV gets inside a macrophage, hides inside little compartments called vacuoles, and just hitches a ride. It uses the macrophage as a Trojan horse to travel anywhere in the body, even crossing the blood-brain barrier.
Which gets it right into the central nervous system, yeah.
But wait, I have to ask, if the body's immune system is a highly trained security force, literally designed to detect and kill invaders, why doesn't it just wipe out these infected macrophages immediately? How is this security system letting an infected cell just cruise right through the bloodstream?
Because the security system is completely blind to it.
Blind to it.
Yeah. That is the terrifying genius of a latent infection. The human immune system detects foreign proteins, the actual physical debris a virus produces when it's actively replicating.
Oh, I see.
But during latency, the provirus isn't making any proteins. It's just a silent string of DNA that looks identical to the human DNA surrounding it. So to a passing security guard, that infected macrophage looks like a perfectly normal, healthy citizen.
Wow. It's a sleeper agent.
Precisely. And this latency is exactly why HIV is nearly impossible to eradicate with standard antiretroviral drugs once it hides in those long-lived resting CD4 positive cells. Our medicines target active replication, the machinery of the factory. They can't target silent DNA.
So it just waits. Okay, so if this thing is just sleeping,
Yeah.
how do we get from a silent microscopic invasion to a full-blown systemic collapse? What does that timeline actually look like?
The war essentially plays out in three distinct phases. You have the acute phase, the chronic phase, and finally the crisis phase.
Let's start with the acute phase. That happens fast, right?
right.
Just 3 to 6 weeks after that initial infection.
Right. And it often feels just like a bad case of the flu. Fever, swollen lymph nodes, maybe a rash. Clinically, this is called seroconversion.
But under the hood, the viral load is spiking massively. Millions of copies of the virus are flooding the blood, and those CD4 immune cells take a huge initial hit.
They do. But the immune system isn't defenseless during this initial breach. It mounts a really fierce counterattack. Cytotoxic T cells step in, the viral load drops significantly, and the CD4 cell counts actually rebound and stabilize.
So to the patient, the flu-like symptoms just disappear. They probably think they just fought off a tough bug and are perfectly fine.
Exactly. But they aren't. Because now we enter the chronic phase, also known as clinical latency.
And this is the longest part of the timeline, right? This can easily last 7 to 10 years.
Yeah, a very long time. On the outside, the person looks and feels completely healthy. But inside, specifically deep within the lymph nodes, there is a massive, silent cellular war taking place.
Just grinding away.
Right. The virus is systematically destroying CD4 positive T cells, which are the master coordinators of the entire immune system. And the mechanisms of that destruction are multi-layered and just ruthless.
So how is it killing them if it's latent?
Well, some of it is active. First, you have direct cytopathic effects, meaning the virus actively replicating inside a cell simply bursts it open and kills it.
Right.
But the real devastation comes from apoptosis, which is programmed cell death. The constant lingering presence of the virus puts the entire immune system in a state of chronic hyperactivation. It's so overstimulated that even uninfected T cells get the biological signal to self-destruct.
Wait, really? So the immune system is getting so stressed out by the constant alarm bells that perfectly healthy, uninfected cells are just committing suicide?
Yes, the collateral damage is immense. And HIV also causes something called syncytia. This is where an infected cell binds to healthy uninfected cells, fusing them all together into a giant ballooning mass of useless cellular material that dies within hours.
Oh, wow. So it's taking out surrounding bystanders just by touching them.
Exactly.
Reading the mechanics of this, I couldn't help but picture two massive armies fighting on a battlefield. For years, one army, the human body, is replacing its fallen soldiers exactly as fast as they die. It's churning out new CD4 cells every single day to replace the millions being slaughtered in the lymph nodes.
A war of attrition.
Exactly. From the outside, it looks like a perfect stalemate. But eventually, after a decade of nonstop combat, the body simply runs out of reinforcements. The bone marrow and the thymus just can't keep up the production rate anymore.
And that exhaustion marks the transition into the final stage, which is the crisis phase, AIDS.
And clinically, there's a very specific definition for that, right?
There is. We define AIDS by a specific metric regarding those CD4 cells. A healthy person has a robust ratio of CD4 to CD8 cells, about 1 to 2. When you reach the crisis phase, that ratio flips to less than 0.5. Or, more simply, your CD4 count drops below 200 cells per microliter of blood.
At that point, the master coordinators are just gone. The generals have left the battlefield.
Right. The immune system has essentially collapsed. And it's vital to understand here that HIV itself doesn't actually kill the patient directly.
It doesn't.
No. Instead, it systematically dismantles the bodily security system, leaving the front door wide open for opportunistic microbes that a healthy body would normally brush off without a second thought.
Right, because once the CD4 cells are gone, these organisms replicate completely unchecked. And hold on, I want to talk about these opportunistic infections because the reality of them is chilling. We're talking about things that are just floating around our everyday environment, suddenly becoming fatal.
Precisely. You have things like Toxoplasma gondii, which is a pretty common protozoan, suddenly causing severe brain abscesses.
Wow.
You see severe fungal infections like Candida causing debilitating thrush, or pneumocystis causing a really deadly form of pneumonia. And bacterial threats, most notably Mycobacterium tuberculosis, become incredibly aggressive.
Pathogens that just rely on the host being immunocompromised finally have free rein. So seeing the utter devastation this virus causes, how it operates on this invisible timeline, it naturally raises a massive question.
Which is?
Well, if we've mapped its entire genome, and if we know every single step of its life cycle attachment, fusion, reverse transcription, integration, why don't we have a cure or a vaccine yet? I mean, we've cured other viruses.
It remains one of the greatest frustrations in modern virology, honestly. Developing a vaccine for HIV is uniquely challenging primarily because of its mutation rate.
Because it's sloppy.
Extremely sloppy. HIV replicates very poorly. It makes constant mistakes when copying its genetic code, and those mistakes result in constant mutations. So by the time the immune system figures out how to attack one version of the virus, it has already mutated into a completely different shape.
You're trying to hit a target that changes its appearance every time you aim at it. But here's where it gets really interesting, though. Because it's not just mutating, right? It's actively hiding.
Oh, the NEF gene. Yes.
Yeah. There's this specific viral gene called NEF. And from the notes, this NEF gene actually forces the infected cell to pull its CD4 receptors back inside itself. It removes the flags that would normally tell the immune system, hey, I'm infected, destroy me. It acts as a literal biological cloaking device.
The virus is actively sabotaging the cell's ability to call for help. It's brilliant in a dark way. And the most ironic, perhaps cruelest twist is the fundamental nature of vaccines themselves.
How so?
Think about how a normal vaccine works. It stimulates the immune system by waking up and activating CD4 positive T cells so they can learn to recognize a threat.
Okay.
But what exact type of cell does HIV specifically target, infect, and destroy?
Activated CD4 positive T cells. Oh my God.
Exactly. By trying to provoke a strong immune response with a traditional vaccine, you are inadvertently providing the virus with an all-you-can-eat buffet of exactly the type of active, hungry cells it needs to replicate and spread.
That is just diabolical. You try to arm the soldiers, and the enemy literally feeds on them. But it's not all doom and gloom in the research, right? There are incredible beacons of hope being developed right now, like the VSV HIV vaccine platform.
Yes, that approach is brilliant.
It really is. They take a completely different, harmless virus, the vesicular stomatitis virus, or VSV, and genetically engineer it. They strip off its normal outer coat and dress it up in the GP120 envelope of HIV.
Right, because the VSV virus is what we call replication competent, meaning it multiplies safely in the body without causing any disease.
But because it's wearing the HIV envelope as a disguise, it safely trains the body to make specific antibodies, and it activates killer T cells against that exact GP120 shape.
Precisely. It primes the entire security system without ever introducing the actual danger of HIV's genetic payload.
It's like running a high-stakes fire drill, using a very realistic hologram of a fire. No one actually gets burned, but everybody knows exactly what to do when the real thing shows up.
That's a great way to put it.
But for people who are already infected, since we don't have a cure yet, the counterattack relies on Antiretroviral Therapy. And this isn't just one magic pill, is it?
No, not at all. It is a very specific cocktail of drugs. And a combination approach is strictly necessary because of that sloppy replication and high mutation rate we discussed earlier.
Right, because if you use just one drug,
the virus will rapidly mutate a resistance to it and just bounce right back. But if you hit it with 3 or 4 different drugs simultaneously, targeting completely different stages of its life cycle, the virus mathematically cannot mutate fast enough to survive all of those roadblocks at once.
that makes total sense. And instead of throwing a bunch of complex chemical names around, let's look at what these drugs actually do. You have inhibitors that jam the reverse transcriptase machine, stopping the virus from turning its RNA into DNA.
Right, cutting off the translation.
Then you have other drugs that act like glue in the protease factory, stopping the virus from cutting its newly made proteins into usable pieces. So you hit it at the front door, you sabotage the factory floor, and you barricade the exit.
And if we connect this to the bigger picture, the success of ART is absolutely staggering. I mean, it suppresses viral replication so effectively that the viral load in a patient's blood drops to completely undetectable levels.
Which means they can't transmit it either, right?
Exactly. Undetectable equals untransmittable. This preserves the remaining CD4 cells and completely halts the progression to AIDS. It's vital to note it is not a cure. It cannot touch that latent provirus hiding in the DNA. But ART has transformed what was once a fatal crisis into a manageable chronic condition.
Unarguably one of the greatest medical achievements of our time.
Without a doubt.
It truly is. And the science isn't stopping at just managing the disease either. There are experimental frontiers looking at how to actually clear it out completely. Scientists are looking at certain cancer drugs, like PEP05, combined with other compounds that could do something normal. ART can't.
The shock and kill approach.
Right. They're finding ways to actually attack those dormant HIV reservoirs. The goal is to wake up the sleeper agents, force the provirus to go active so the standard drugs can finally see them and kill them.
Which is just incredible. We might be a decade away from human use, but the fact that we're figuring out how to smoke them out of the DNA is amazing. It's the next logical frontier in virology. If we can activate the latent reservoirs while simultaneously suppressing any new replication with ART, we move from just managing the disease to potentially eradicating it from the host entirely.
So what does this all mean? We started this deep dive talking about how people assume they understand HIV and AIDS. But looking at the cellular mechanics of this virus, you realize it's not just a medical condition.
No, it's a marvel of evolutionary biology.
Exactly. A microscopic stealth submarine that steals our own cellular membranes, rewrites the fundamental rules of genetics, cloaks itself from our security forces, and systematically exhausts our bodily defenses over a decade. But against all of that, human ingenuity has found a way to freeze it in its tracks. We've mapped its genome, we're building decoy holograms for vaccines, and we're developing ways to flush it out of its hiding spots.
It really highlights both the fragility of our biology and the profound power of the scientific method. We're fighting a war against an enemy that integrates into our very being, and step by microscopic step, we are finding ways to win.
We really are. And speaking of integrating into our very being, I want to leave you with a thought that really stuck with me after exploring all of this. We talked heavily about how HIV as a retrovirus uses integrase to perfectly splice its genetic code into human chromosomal DNA, where it can hide for a lifetime as a silent provirus. Well, millions of years ago, our ancient ancestors were infected by other retroviruses, and some of those infections occurred in reproductive cells, meaning those integrated viral codes were passed down to the next generation and the next and the next. So if a virus can perfectly splice itself into our DNA and just stay there, how much of the human genome inside you right now is actually just ancient dormant viruses that infected our ancestors millions of years ago and never left?